So welcome back to Virtual March of Dimes High School Science Week. If this is the first time you are tuning in, my name is Madison Dodds, and I'm a member of the Salk Institute's Education Outreach Team. This event is a Salk tradition. This year is the 31st annual event taking place, and while we wish we could have you on campus with us, we are proud to continue this event in a virtual space. It's something that not only our team, but Salk researchers look forward to each and every year, and they're eager to interact with you throughout this week. So the Salk Institute was named after Dr. Jonas Salk, and you may have heard his name recently in the news, as he was a key player in another public health crisis, the polio epidemic of the 1940s and 50s. Dr. Salk developed the first safe and effective polio vaccine, and then he went on to pursue another dream, which was to create a collaborative environment where researchers could explore the basic principles of life. Founded in 1960, nearly 60 years later, the Salk Institute is a world-renowned basic biological research facility. So this is a bird's eye view of the courtyard that you saw in the last couple photos. And something that's really unique about the Salk Institute is it's actually a 6-story building, but because of coastal regulations, the first 3 floors are actually underground. So in this picture here, you can see some of these light wells, which allow natural light even on the first floor, which is 3 floors underground. So it's a really nice place to work. Nobody's stuck in the basement. Everyone has access to natural light. As you walk up to the courtyard, you might notice that there's these plants over here on the left-hand side of this image. This is actually an orange grove that produces fruit, and Salk employees, they'll harvest them on a regular basis, and everybody can take them home and enjoy them. It also smells really nice when they're in bloom and you're walking up to the courtyard looking at the Pacific Ocean. And I don't know if you're like me, but I really like symmetry, so this photo really makes me happy. So a little bit about today. So today you are going to have the opportunity to participate in a virtual lab tour with the Molecular and Systems Physiology Laboratory under the direction of Dr. Janelle Ayres. We're going to start with a little bit of background about the program and some other opportunities we have for high school students. Then we will air the virtual lab tour video, and it will be followed by a panel discussion. And we'll end about 1:25 with some closing remarks. So each lab tour is a standalone experience. You're welcome to attend only those that align with your interest or all of the events. It's really up to you. We definitely recommend checking out at least a couple of the events because you might be interested in a field you've never considered before. Each lab tour does have a separate link, especially if you're planning on joining us for 2 events in the same day. Please make sure you're using the correct link so that way you can participate. While we are very hopeful the event will run smoothly, if technical issues do arise, please be patient. And if that happens, please feel free to email education@salk.edu for assistance. Our favorite part of this week is really interacting with you. We want to know what you're curious about. We're going to be using student questions to guide our panel discussion. So if you would like to submit a question, either maybe something you saw on the website, or you're curious about something featured in the video, or something our panelists said, we'd love to hear from you. So to submit a question, go to the video description and click the link for the form. We thank you so much for submitting questions. We're going to get to as many as we can. If you'd like to read a little bit more about the lab's research or get to know some of our panelists, click the Show More button, and you'll be able to see a link to the event's webpage with all this information. You're welcome to submit a question for an individual panelist or for the entire lab. So if you are excited to be here this week and are looking forward to other opportunities for high school students, I'm happy to share a couple that we have. So the first is the High-Tough Brody High School Summer Scholars Program. This is our 8-week internship program. The first week of the program is a biotech boot camp, which is a crash course in all things biotechnology to help you learn some of the techniques that you would use in the lab. It's then followed by 7 weeks in the lab paid, where you work with a mentor that's a Salk scientist, and you are going to conduct real-life research and build some science communication skills to help you be successful in future academic and STEM settings. You also have the opportunity to participate in other outside events like industry visits to local biotechs, seminars, and other fun things to do to get to know your fellow interns. While we are very hopeful the program will take place in 2021, it is dependent on the public health recommendations at the time. So please check out our website later in March for updates on the program status. So I talked a little bit about the science communication skills that the students build throughout the program. So in this picture on the top left, you see all of our former interns all dressed up, ready for their scientific presentations, where they were able to present to an audience of their labs, their friends and families about what research they had done that summer. Now, if that program isn't able to take place, or even if it does, we do have another virtual program that is not limited to San Diego County students, and that's the Introduction to Research Science and Communication virtual program. This is a 5-week professional development program for students. It's about 5 to 10 hours a week and mostly asynchronous. We took elements of the High-Toff-Brody High School Summer Scholars Program and developed short sequences to help you build skills to be successful. The elements include reading scientific journal articles, analyzing data, preparing a scientific presentation, and virtual labs. Throughout the summer, you have the opportunity to work towards a capstone project, which is a brief research project on a topic of your own choice, something that you're interested in. This program will take place summer 2020, and the lottery form to be considered to apply for this program is currently found on our website. The deadline is on March 31st, so make sure to apply if you're interested, especially if you're joining us from outside of California. This could be a great opportunity to learn more about Salk and to build some cool skills. So I went pretty quickly through everything. That's quite a few of our high school programs, but we really want to make sure that you know how to reach us if you're curious or have questions. Please feel free to contact our team at education@salk.edu or go to our website, www.salk.edu/education. You can also find some frequently asked questions pages here if you want to know more. Now, this event is not possible without the generosity of our sponsors, so we would like to say thank you. The March of Dimes High School Science Day is made possible through the generous support of the Anne and Neil Blue High School Science Fund. Too many S's in a row. Our sincere thanks to the Blue family and all the supporters, partners, and volunteers that make education outreach possible, including the March of Dimes. So today, this is one of my favorite parts of this event. It is my pleasure to introduce the virtual lab tour featuring the Molecular and Systems Physiology Laboratory of Janelle Ayres. The video gives you a glimpse of what a day in the life of the scientist can look like. Each video was designed and filmed by the scientists themselves, so each one will be unique. The videos will highlight some technology and tools that our scientists use, but also introduce you to the people that work there and some potential career options if you're interested in going to a STEM field. It is my pleasure to introduce the Molecular and Systems Physiology Laboratory of Dr. Janelle Ayres.
Hello, my name is Robert. I'm a grad student in the Ayres lab where we study infections. And typically when you think of your body dealing with an infection, you think of it using your immune system to kill the pathogen. But we study this thing called cooperative defenses, where you can actually promote health without killing the pathogen.
a graduate student in the Ayres lab, and I study how fat can help with a parasitic infection and maintaining health during an infection. And so today I wanted to show you a technique we use in the lab quite often called qPCR. So this is actually a technique that's used to measure the amount of genomic material, so RNA or DNA present in a sample. You may have actually heard about this in the news lately because this is the same technique that doctors can use to determine if somebody is infected with SARS-CoV-2. So the idea just being if you have genomic material such as RNA from the virus present, then you actually have the virus. So I'll show you how we do this in the lab. Adding DNA from samples that I extracted previously, so in this case my parasites, to determine how much DNA is present in each sample in different conditions. I'm adding Cyber to the plate, and Cyber is a reagent that we use that has a dye in it that actually fluoresces with the DNA, so that helps us measure the amount of DNA there. So now I'm putting the plate into the actual qPCR machine, which is the large white block off to the left. So this machine just cycles through different temperatures and actually allows for the qPCR to amplify the genomic material. And so there's actually a computer on the other side that controls this machine, and I input all of the temperature changes that I want it to go through. So these are some of the results that you'll get from qPCR. So this is looking at a very specific gene that we just put into the machine, and these lines here that are going up kind of exponentially and then leveling off are different samples. And so the idea behind this qPCR is you have these cycles, and every cycle you're doubling the amount of genomic DNA present until it reaches this threshold where we can detect it. And so any sample that crosses this line, we then say it's detected at this point. And so the reason this can then quantify the amount of genomic material is that every time you go through a cycle, you're doubling the material. So I have some samples that I put in that are my standard. So I know how many, in my case, parasites, but in the case of COVID, you know how many viruses there were that you extracted DNA from or RNA from and put it in. And then if one of your samples came up one cycle later, you know that it started with half as much genomic DNA or half as much virus because it had to go through one full more cycle to reach this threshold. And so these are the results that you get from the qPCR that I ran, and then this is exactly what you would expect in a clinical setting if you're testing for COVID.
In order to study bacteria, a standard technique we use in the lab is to grow bacteria on nutrient agar plates.
Hey, my name is Grishka. I'm a microbiologist in the lab, meaning that I study bacteria and viruses. My research focuses on how gut pathogens cause disease and how these pathogens evolve over time during infection.
Here, I'm demonstrating how we grow bacteria for experiments. Using a sterile toothpick, I can transfer bacteria from an old plate to a fresh agar plate. I streak several times in order to isolate individual colonies of bacteria. Next, we put these plates into 37-degree incubators. E. coli grows very fast, dividing every 20 to 30 minutes, meaning that overnight we'll get a full plate of bacteria. These particular bacteria that I grew up express a red pigment, meaning that when we expose these bacteria to UV light, they actually fluoresce this red color. These bacteria are very useful for our experiments because we can use this red fluorescent protein to visualize bacteria during infection.
7, 6, 5, 4, 3, 2, 1.
I'd like to welcome some of the panelists from the Ayres Lab to thank you so much for showing us a glimpse of what a day in the life of your life is like. Let's take a moment to do some introductions. So if I could have all the panelists say your name, where you're from, and then what your role is in the lab. So let's start with Grishka.
Hi, so my name's Grishka. I'm a postdoc in the lab, so that means that I've already gone through grad school and earned a doctorate. I'm originally from the Bay Area, but now we're in San Diego in La Jolla. So my research, I am interested in bacterial pathogens and how they cause disease, and I'm particularly interested in trying to understand how these pathogens evolve during infection.
Awesome. Thank you for joining us today, Grishka. I think we do have some schools that have registered from the Bay Area, so we have a hometown point for you. All right, and Sam, what about for you?
Hi, I'm Sam, and I'm originally from Hudson, Wisconsin, so a small town in Wisconsin. And I'm a graduate student in the lab, so I'm working towards my PhD currently, and I research a parasitic disease and how adipose tissue can help during an infection.
Yeah, and can you just tell us a little more? What's adipose tissue?
Oh, so fat tissue, and kind of there's a lot of immune cells in fat, but we always think of it as just kind of storage for energy. And so I'm looking at how that might play a role during inflammation and disease in general.
Awesome, thank you. I've never thought about fat cells having an important role in the immune system, so thanks for sharing. Thanks for joining us. All right, and Sarah.
Hi, I'm Sarah Stengel. I'm a research assistant in Ayres Lab, and I basically assist our PI, which is our principal investigator, Dr. Janelle Ayres, with her research, basically. And I do a lot of like molecular biotechniques like qPCR and Western blot and stuff like that. And I grew up in San Diego. I went to Canyon Crest Academy, in case anyone's here from that.
Yep, they are registered, so shout out to Canyon Crest.
Shout out Mr. Quinn. Okay.
Alrighty, so let's dive in. And for our first question we have that's been submitted by a student is, how did you know being a scientist would be a good career choice for you? I think a lot of students are kind of trying to figure it out at the high school level, so we'd love to hear how you know it was a good choice for you. So Sarah, would you mind sharing first?
Yeah, so I was one of those people that as a kid, I always kind of loved biology and I just found it really interesting. So in high school, I took like bio classes like we needed to, and I thought they were really cool. And then in college, I kind of gravitated towards them. And so by the time I finished college, I was like, oh, I want to make a career out of this and just kind of follow that path of like anything science related. I found super interesting.
Awesome. What about for you, Sam? How did you know this would be a good pick for you?
Yeah, I would say my interest first started in biology in general when I was really young, growing up kind of in rural northern Wisconsin. I got really interested in kind of just nature and ecosystems because there's a lot of lakes and forests and really neat biology up there. And then I actually went to high school in South Dakota and worked with corn breeders. And it happened to coincide with when I first started learning about genetics in high school. And it was really cool to be able to see the application of this like textbook science in class, and then be able to see like how this is put into effect like in the real world. And so that made me really want to start studying science and applying it.
Yeah, I know that's something a lot of students can relate to. It's always wanting to know, why is this important? Why does this matter? So being able to see what you're learning in the textbook really come to life is, I'm sure, was a pretty impactful experience. And for you, Grishka, how did you know this would be a good career choice?
I think it's pretty similar to what Sam and Sarah said. Like in high school and in college, I always gravitated toward science, and in particular biology. Had the most fun in those classes, and particularly like lab courses and like some field study. I mean, my suggestion actually to high school students who are thinking about doing science as a career, I think if you like science, if this is one of the most interesting classes or courses that you take, while you're in school. I mean, just get your hands dirty and try to, join a lab or work on some project that's science related. As a career, there's actually a lot of different science careers. In particular, we do more research focused, but I think there's space for everybody. If you're thinking about doing education or policy or health. So, I mean, if you like science, you just go for it.
So you guys all kind of touched on a little bit about how being in high school, it did have a form of impact on what you wanted to do as a career. Now, maybe something you'd learned in science classes or just in general, what are some skills that you learned in high school that are benefiting you now? So maybe Sam, you could tell us if there's anything you'd kind of learned in high school that's benefiting you now?
Yeah, I actually think for like my current career as a graduate student, that's just learning a lot of like basic writing skills has been the most beneficial for me, which is not something I expected necessarily going into science, but a lot of whether it's graduate school or whether you're a postdoc, you have to write very clearly what you're thinking for what your science means and explain it to others. And I think just having a foundation where you just know good communication through writing has been really important.
Yeah, especially when right now, when we're in this virtual world where we're not getting that same face-to-face time, I think writing is especially important because a lot of the written communication is done via email. So being able to express yourself clearly is going to become more and more important as we kind of transition to this hybrid situation. What about for you, Grishka?
One thing that I think that just comes up like every single day is just math. You need to be really comfortable with math. We do a lot of measurements and weighing things. It's nice to be able to do like math really quickly in your head. You don't have to be we don't do very complicated math, but math is something you should be really comfortable with.
I saw a lot of nods from the panelists when you said, as soon as you said math, everyone's like, yeah. What about for you, Sarah? What did you learn in high school that's benefiting you now?
One thing I thought was really important that has helped me a lot in life is I don't know if it was my high school atmosphere, but every teacher was really welcoming and inclusive, basically. So I felt comfortable building relationships with my high school teachers, which kind of carried over into college, which became extremely beneficial because I got to join multiple labs because I had no problem talking to the professors like normal or asking them questions or not being so scared. And then nowadays when I have career questions or I don't know what path to take, or I might have want to enter an industry, I can easily email them or call them or text them even and be like, what's your advice on this? And they have no problem talking to me basically.
I think sometimes it's just making that first initial communication. You feel nervous, but almost everyone is going to be more than happy to help you and to step in and to provide guidance. I think people are always happy to help. Mm-hmm. So we have another question submitted today. It is, what was your experience like when you first stepped into the Salk Institute, and what has made you stay? So let's hear from Grishka. What was your experience like?
So Salk is actually pretty different from other places I've been. So I've mainly, before the Salk, I was doing research as an undergrad, as a graduate student in the university setting. So there are a lot of undergraduate students around a lot of labs. Salk is a very, it's a small institute. It's very tight knit. The culture is actually very different, I think, compared to the other labs that I've been to. It's very competitive here and really fun. And as a postdoc, this is not a permanent position for me. I've been here for over 3 years now, and I really enjoyed my time here, and you know, as a scientist, we actually travel a lot. Like we move around a lot.
It's like an added perk. Do you want to share just a couple cool places that you've been? Maybe not recently with the pandemic, but is there any cool opportunities that you've had as a result of your career?
Uh, you know, I mean, in terms of school, like I grew up in the Bay Area, and I ended up in Wisconsin for grad school. But you know, as a scientist, we travel around a lot for like research conferences. So I've been to Chicago, to Boston, to New York, to these big conferences to uh, talk science for days.
Another reason why science communication is so important, kind of like Sam was talking about, it's so important to be able to communicate science, not just written, but also to an audience of your peers and present. What about for you, Sarah? What was your experience like when you first stepped into Salk, and what's made you stay?
I really loved it. Like everything Grishka was saying, the atmosphere is really cool and welcoming, and especially our lab, it was a lot more, I wasn't expecting how like fun and kind of like goofy you can be. It's like not as serious as I thought when going. Like I graduated UCSB with a bachelor's, and I just for some reason got it in my head that like a research institute would be more like serious. But as soon as I joined our lab, I realized that like we all kind of are figuring out like what we're doing, and everyone's like really supportive and helpful and nice.
I think that really speaks. It's not only important to find a place where you like the research, but place where you feel comfortable to be able to really grow your own professional skill set. And I think your lab you could really tell in your video how close you all are, and what a nice environment it is for all of the scientists. And for Sam, what was your experience like stepping into Salk?
Well, I kind of like laughed a little bit thinking about this in my head because my first time stepping onto Salk was during my interviews for graduate school. And so just for some context, I was in undergrad at Minnesota. And so I flew out of Minnesota and it was negative 20 degrees, and then stepped onto Salk campus, which is super pretty and it's sunny and like 60. So that was a very good first impression. But otherwise, what made me stay is kind of what the other two panelists were saying, is just I really liked the lab culture of Janelle's lab, and joined specifically for her lab then at Salk. And then also, I'd say I was really impressed with Salk is kind of like an open lab policy. So like there's no dividing walls or anything between labs to kind of foster communication between labs. And I really like that because you end up walking into like a neighbor's lab to ask them questions in their specialty. And so you just have a lot more collaborations, which I really liked.
That kind of leads us perfect up for this next question, which was, how does teamwork play a role in your research and your lab's work? So since you already kind of were starting to allude to that, Sam, can you talk a little bit about that?
Yeah, absolutely. I would say it plays a huge role just because, you're like, as a graduate student, I'm working to become like an expert in my subject matter, and really narrow down my research interests. But to kind of figure out what you want to do next and kind of connect to other fields, you're constantly trying to learn like new information, but you can't be an expert on everything. And so it's really helpful to just have people around who are experts on other things that you can go ask them questions on, whether that's within your lab, like if I have a question on microbiology, I'm going to go ask Grishka, or if it's something between labs, like we have a metabolism lab right next door that I do a lot of work with. And it's a huge benefit to just be able to go walk and talk with them, or have them help with like a new assay or something.
Awesome. What about for you, Sarah? I know you talked a little bit about how you work closely with Janelle, your principal investigator, but what about for the other lab mates? Do you have an opportunity to collaborate and work with other labs?
Yeah, so when I first got hired, I didn't know how to do like qPCRs or Western blots or anything like that. And then, so you kind of pick up on things in lab by your other lab mates. Like Janelle will be like, talk to so-and-so and learn how to do it. And so you're constantly, at least me, are like working with other people to learn new techniques or how to do them, or like troubleshooting. And stuff like that. So it's really open and it's really nice because if you fail or anything like that, you can just, I feel totally comfortable going to my like coworkers and asking them like, has this happened to you? And if so, like, what'd you do? And that kind of thing.
Yeah, learning from each other is so, especially when you can take their knowledge and use that to grow faster, it's so helpful. It's nice to have that collaborative environment. And for you, Grishka, as the microbiologist on the team, what is it like for you to collaborate?
So, the thing about doing research is we're very knowledgeable about like very narrow piece of the world. So a lot of times, as Sam and Sarah were talking about, I end up going to a lot of times going to talk to other people to try to probe their mind, get their expertise. So this is a very common thing. I mean, one thing that I really enjoy about our lab and other labs that I've worked on is, we essentially like mentor each other. So like when I was starting out, I would look to grad students and lab managers to help me do my work, and hopefully I'm 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 passing it down. I'm giving some knowledge to other people in my lab. So I mean, the teamwork, mentoring is a really huge part of science.
I think that's a really important point because a lot of times, like when you think of like the big science awards, like the Nobel Prize, it's like credited to like one or two people, but really all the people in the lab that really are making these discoveries possible, it's really a team effort, and collaboration is just so critical. All right, so what is the most interesting part of your job? So Sarah, what about for you? What is the most interesting part?
I might brainstorm on this, so you can go to the guys. I'll think.
Okay. All right. For you, Grishka, what is the most interesting part of your job?
The thing I like most about research in general is just, and I'm sure people have heard other people talk about it. It's, there are specific moments where you're the only person in the world that knows something, and it's some minute fact, but that's what I enjoy the most. Um, yeah, I can. I need to think.
That's
What
okay.
other?
Sam, is there anything that's jumping out at you as like, this is the most interesting?
Yeah, I would say, I mean, what Grishka said is true too. You're the only person that knows something. It's kind of a cool feeling. I would also just say like a large part of your job learning new things. And we were saying you become an expert in a very narrow field, but you still get a lot of benefit from like learning things from like random fields. Like famously, our boss Janelle, like her research actually stemmed from plant research, where she was reading some papers in like about well-established things in plants, and she had the idea of maybe our immune systems kind of act in a similar way. And so I really enjoy that aspect and find it super interesting. Is just like sometimes I'll skim a paper that has nothing to do with my research, but just something that interests me, like fungal biology or something. And I still think it helps contribute to my job and is like a part of it.
Yeah. And for me at Salk, I think the most interesting part of my job is getting to interact with scientists like you. I learned something, like even today, like learning about how fat cells can play in a role in the immune response. That instantly sparks questions for me. So now I'm going to go home and do some more research on that. I just love being able to be at Salk and have that curiosity is always, that flame of being a learner is always kind of stoked. For Sarah, is there anything you want to talk about like that's most interesting, or just kind of build off what they said?
Yeah, I agree with everything they said, but also one thing I found really interesting was I finally get to see like real world examples of things that I learned in school. So I remember learning about some of the stuff in my college courses, like Red Queen hypothesis and stuff. And I get to see all these like different things I learned in school basically applied to like things we keep finding.
That's so cool. Yeah, I think that's definitely like bringing it to life for me. Anytime I see those images of the fluorescence, so that you had in your guys' video of the bacteria, and when you get to see that like actually under a microscope, I always just think it's like the mix of being art and science at the same time. It's beautiful. So kind of you guys talked, Grishka and Sam, you talked a little bit about like how you can become very narrow in your focus. How did you know that's what you wanted to study in the lab?
Well, I guess for me, it was, for graduate students, you kind of go through a rotation process where you try out different labs. And so I kind of came in with a focus in biochemistry and molecular biology, and was reading, just flipping through lab websites, like finding papers of which labs interested me. Like, do I just find this research compelling? I once had a professor that told me there's a cool factor that you can't really describe. It's just like you think it's cool, and you just go with that. And so that kind of helped me settle on some labs that I tried out, and then mentioned that I found this, like, Janelle's lab culture super great, and I really like the research. And so I joined Janelle's lab, and then for my specific focus within the lab, it was a lot of discussions with Janelle. And so that's kind of where I'm at in the scientific process. Like, I'm learning how to conduct science and come up with my own research ideas. That's what Janelle's kind of like my mentor for that. And so there's a lot of conversations with her about different papers, what is possible to study, like from what's known, and kind of narrowing in on what to research from that.
Yeah, I think having that mentorship aspect is really nice, and having that intrinsic motivation of something that you just enjoy. Sometimes that's how we kind of fall into it. For you, Sarah, was it kind of a similar experience? Like it just felt cool, or is there anything in particular that drew you to this field of study?
Yeah, so when I was in college, I actually got my Bachelor of Science in zoology, so very different. But during my time, I actually took a couple infectious disease and non-infectious disease courses, and I thought they were super interesting, learning about like zoonotic diseases, like malaria and stuff. So I was looking for an infectious disease lab when I moved back to San Diego after school, and that's how I found Janelle's lab, and I've loved it ever since.
Yeah, I think in college it's so important to kind of take a variety of different courses because you never know what's really gonna really spark your interest. But that kind of sounds really cool. What about for you, Grishka? How did you know you wanted to pursue a microbiology career or research?
I think Sam's right. There was just a cool factor to it. It was in college. I took a lot of different science courses, and I always went back to microbiology for some reason, and I ended up working in a microbiology lab, and I didn't want to stop. That's essentially how I got started and kept going. Um, I wasn't actually even particularly interested in infectious disease before. I'm interested in a lot of different aspects of microbiology, like ecology, um, you know, uh, biofuels, all that. And it just happened that, once I got to grad school, I clicked with a specific mentor, and he was working on infectious diseases, and I wanted to continue. And that's what brought me to Salk, because I wanted to uh, look specifically at infectious disease and physiology, and Janelle is an expert in that. So that's why I joined.
Find yourself a good mentor. Find something that's like that you're passionate about. I think that's like great advice that you really can't go wrong, regardless of whether you want to pursue a career in science or in another field. So I think something a lot of students can be concerned about is failures or errors in your experiment. So the next question kind of has to do with what happens when stuff goes wrong, which is, as we know in science, is quite often. What happens when an experiment fails? Is there a process you go through to figure out what happened and why that happened? So I'll kind of leave that open for any panelists that really kind of some things jumps out.
I mean, I can go. You just do it again. You I mean, I mess up all the time. I've had really big experimental fails before, and
Raise your hand if you've had an experiment fail. It's like everybody. Yeah.
all the time. I remember in grad school, there was when I just started in the lab. There was an experiment I worked on for 2 months, and by the end of it, I realized I messed up in the beginning. So I had to redo the experiment again. Um, it's all right. You know, it just happens, and you just repeat it. It's part of the scientific process.
Yeah, we learn more from failures than we do from success sometimes. What about for you, Sam?
I would say also one of the things I've been trying to work on, like when a experiment doesn't work, whether it's like I mess something up at the beginning or it just doesn't work from like the way I expected it to, is still trying to get like some usable data from it. So it's still like an ability to or an opportunity to like learn something new. So like I had a lot of experiments that didn't work like around 6 months ago. And it was just kind of like looking at each of them individually, they looked like they didn't work. But then when I looked at them all together, I was able to figure out new directions for my research to go in. And so you're always going to have like experiments fail or not work out, but there's still usually usable data from those experiments that could give you new directions and could lead you to even more exciting avenues of research.
And how do you stay motivated when after a failure that occurs? Or like, especially if when Grishka, it's like 2 months later, you have to realize you repeat. Like, how do you stay motivated in those situations to continue on?
Well, in that case, I had a committee meeting in a month or so, so I had to present something. So I had to push through it. You know, it can be demoralizing, but you know how I look at it is, yeah, everything you do is going to be a learning experience. You're gonna do it better the second time or the third time, and you know, also just I usually try not to expect a specific result or anything. I'd like to be surprised, you know, at the end. Um, that's being like enjoying the work and being surprised by results is what motivates me a lot of the time.
What about for you, Sarah? Do you have anything to add about what you do when an experiment fails and your process after that happens?
Yeah, so I just had like a major thing fail. I was basically running qPCR plates, like the thing you guys saw in the video, for a couple months, and the basically data we got at the end wasn't looking great. And I kept tweaking things over weeks and weeks and running plate after plate and working with Janelle and basically just trying to figure it out. And we found out it was actually like the double DNA binding dye, basically, that was like faulty. And so that was a lot of time and a lot of money. But in the end, now I know what to kind of look for more. And she never got mad at me. Like, I feel like your PI won't get like angry at you if you're trying your hardest and not like intentionally ruining anything. Like, they're really understanding. And so she and I just like talked, and it was not a big deal.
I feel like in science, you have to be like passionate and about like, the scientific method, but also problem solving and like thinking through like backwards, like what's the first thing that could go wrong, and testing every single step, which is kind of like engineering. So it's really that mix of science and engineering. So we have time for a couple more questions. So we talked a little bit about some of your guys' educational backgrounds, but what did you focus on studying in school? And maybe talk about in high school, any classes that impacted you, and then any standouts in college or grad school. And then feel free to take anybody who has a class that sparks out.
I can go. In high school, my favorite class was actually an ecology class, because we actually had a semester-long science project that we designed and performed and actually presented results on. So I remember what I did was doing a survey of sand crabs in San Francisco. That was really fun, and got me really interested in biology. In college, I did a lot of biology courses. I ended up focusing on microbiology specifically. But another course that was really interesting to me, like I got really interested in organic chemistry as well, and it can be pretty useful in biology research.
Yeah, and Sarah, you had taken your background you mentioned was in zoology. Like, how did that come about? How did you choose that?
So as a kid, I was obsessed with like pandas. And so I thought I was going to study pandas one day.
Oh.
And so, but it was like I wanted to pursue zoology, but I didn't really figure out classes that I could take in that field or a career path until I got to college. In high school, my favorite classes were like Japanese and chemistry. If Mr. Shakiri is here, he always like set off the fire alarms and blew up walls and stuff. But it wasn't until college where I really figured out like what a zoology class really means, or ecology and evolution and that kind of thing. And I just got more and more interested in learning anything about it. And that's what made me join like labs. And that's what kind of led me to where I am now.
And so, Sam, I know your college background was a little bit different than what you're pursuing in grad school. Can you talk a little bit about what you were studying in college and kind of how that interest was sparked?
Yeah, my undergraduate degrees are actually microbiology and biochemistry. And so it was kind of, I changed majors in college. I initially went in looking at biomedical engineering, but then just took kind of what Grishka was talking about, like take a wide range of classes. Because I took a microbiology like intro class that I just thought was mind-blowing, because it was like this whole ecology class, but stuff you couldn't see. And they're focusing on extremophiles, like these crazy environments that the microbes lived in. So then I declared for a microbiology major. And then as I was progressing through that, I took my first biochemistry class, and I found that just super compelling because it was kind of I saw it as like a way to measure and explain how microbes can impact the macroscopic world. Mm-hmm. And so like different chemicals that microbes produce, and there's so many microbes that they can actually produce enough of these chemicals to do different things, like actually producing like insulin is made by bacteria now that we use as like a medicine. And so then following that, it just was like a natural progression for me to go into infectious disease research, because in my mind, it was still kind of this ecological microbiology, but now in the context of the human body. And then biochemistry is kind of the bridge of like how the microbes are impacting our body.
So kind of tie back to that first question that we had for the students, what comes to mind in one word when you think of the microbiome? So I'll give you a second to think. Anybody brave, willing to go first? What comes to mind when you think of the term microbiome?
I guess for me, it's health, just because the way we research the microbiome is typically kind of how does it affect the health of the host.
So health, okay, that's one. I don't think I saw that earlier on the student responses, so that's interesting. What about for you, Grishka? What's your word that comes to mind?
I would say community. So you can think of it as, microbiome is a community of organisms that interact with each other. I wanted to point out, it's also not just human and associated microbes. There's a microbiome in the soil, in the ocean. There's microbiomes everywhere. But yeah, community.
Community. Okay. What about for you, Sarah? What comes to mind?
I'm going to steal the boys' answers and say community and health. Those are great answers, so I'm going to agree with them.
I think about evolution because I don't think we could have evolved to be where we are today without our microbiomes. And I think it's so the more and more we discover about our microbiomes, the more and more we realize their role in our overall health of not just us, but our planet. So I think that's what comes to mind for me. All right, this is our final question. Time has flown. So our final question is, what is something that you've learned in the lab that has changed the way you live your life? That's kind of a heavy question. So it might be science related, it might not. I'll give you guys some time to digest that. That's a heavy one. Thanks for submitting that, student. And it can also not just be your Salk lab here. It could be just something that you've learned in another lab.
For me, I think like after doing research for so long and being a scientist, a lot of the times when I like hear a fact or hear some antidote, like my mind just immediately goes into overdrive trying to understand, how does, like, what experiment would you do to know that fact? Or like, what's the data? Like I've become like very skeptical, I think. I have a very critical mind now, I feel. So it just becomes second nature now, like questioning facts or what people say, or I don't know if I don't remember if I used to be like that. Maybe I was always like that, but
definitely
yeah,
kind of sparks your mind. I think that's important, not just in science, but just in general, like to consider evidence, to consider viewpoints, biases, data, all those things. All right, anybody else want to share what is something that they've learned in lab that's impacted their life?
I mean, I could go. So I would definitely agree with Grishka. I think that's a really good point. You have so much of like time in lab when you're reading papers to try to understand like what research you should do next. And so much of your work is dependent on the rigor of other people's work that you analyze it very skeptically. And like Grishka said, it kind of becomes a habit for like when you hear something in the news, it's like, well, how would they actually know that? And then the other thing isn't necessarily science specific, but I've always been very into like outdoor stuff. So I think one of the things that's kind of changed in my life is just kind of with my background in biochemistry, and then I helped teach a nutrition class and an actual exercise physiology class. And I think those courses and everything have definitely changed like just how I eat in general, like eating healthier and just the way I live my life in general, like kind of understanding the science of how to get better at running, for example, and like improve endurance has probably just changed how I like approach my training and stuff. But
it's so cool when you can take something that you learn in a paper or from a course and then actually apply it to your life and see it make a difference. It's always a cool thing. For you, Sarah, has there been anything that you've learned in lab that's changed the way you've lived your life?
I feel like I've become more of a hypochondriac, more scared of germs. But also what Grishka was saying, I feel I relate really strongly to that. Like when presented with either like news or something someone's telling me, I just try to like think through why that would be going on or something, especially with like COVID news and everything, trying not to accept it firsthand and kind of thinking about the biology behind it or something like that.
I totally agree with you. After I took microbiology in college, I wanted to wash my hands all the time. After I touched my phone, all those things that you just don't even consider. But I think we're a little bit more cognizant of it now with COVID to wash our hands and to be aware of just our touch contacts. But thank you all so much. Thank you, Grishka, Sarah, and Sam for joining us today, for sharing your insight on your career paths, on your journey, and what you love about science. We are so grateful for you. We really appreciate you developing the video to give us what a glimpse of a day in the life is like. We'd also like to thank your principal investigator, Dr. Janelle Ayres, for allowing us a glimpse inside the lab. We are so grateful to have you all here today. Students, if you want to learn more about the Ayres lab, there's more information on the Salk website, so definitely check that out. Thanks so much, and we'll see you next time. Bye, guys.
All right. Bye. Thanks.